Assalamualaikum everyone, welcome back to my YouTube channel. Let's start the video. We will talk about the thyroid. What is thyroid gland? Thyroid gland consists of two bulky lateral lobes. Basically, thyroid gland has two lobes, which are lateral lobes, two lateral lobes, and then these two lateral lobes are connected by relatively thin isthmus. Isthmus connect these two lateral lobes of the thyroid gland, usually located below and anterior of to the larynx. The thyroid gland develop embryologically from an invagination of developing pharyngeal epithelium that descend from the foramen cecum at the base of the tongue to its normal position in the anterior neck. Basically, they are telling about the anatomy of the thyroid gland. Thyroid gland contain two lobes which are anteriorly located and present below the anterior or to the larynx and relatively contain thin isthmus, and then thyroid gland develop embryologically. It develop embryologically from the evagination from the pharyngeal epithelium, and after that descend into the cecum. Cecum is foramen cecum, at the present at the base of the tongue and normally positioned in the anterior neck. This pattern of descent explains the occasional presence of ectopic thyroid tissue. It will present the presence of ectopic thyroid tissue, most commonly located at the base of the tongue, which is lingual thyroid. At other sites abnormally high in the neck. After that, we are talking about lobules. First of all, we were talking about the lobes, and now we are talking about the lobules. Thyroid is divided into lobules, each composed of 20 to 40 evenly dispersed follicles. Normally, total dispersed follicles are 20 to 40. The follicles are lined by cuboidal to low columnar epithelium. These follicles lined by cuboidal to low columnar epithelium, which is filled by thyroglobulin, and then the iodinated precursor protein of the active thyroid hormone in response to the trophic factors from the hypothalamus. TSH thyroprotein is released by thyrotrophs in the anterior pituitary in the circulation. The binding of the TSH is, to its receptor and the thyroid follicular epithelial cells result in the activation and conformational change in the receptors, allowing it to allowing it to associate with the stimulatory G protein. As illustrated in the figure, the activation of stimulatory G protein result in an increase in the intracellular CAMP levels, which stimulate the thyroid hormone synthesis and release the thyroid follicular epithelial cells, convert the thyroglobulin into the thyroxine T4 and the lesser amount of triiodothyronine, which is T3. T4 and T3 are released into the systematic circulation, where most of these peptides are bound to circulating plasma proteins such as T4 binding globulin for the transport to peripheral tissues. The binding protein maintains the serum unbound free T3 and the T4 concentration within the narrow limits while ensuring the hormones are radially available to the tissues. In the periphery, the majority of the free T4 is deontinated to T3. The larger binds to thyroid hormone unclear receptors in the target cell, which fold with tenfold greater affinity than T4 and has proportionally greater activity, binding to the thyroid hormone to its nuclear thyroid hormone receptor, which is TR, create a hormone receptor complex that regulates the transcription of the subset of the cellular genes This produce diverse cellular effects including the carbohydrate and lipid catabolism and protein synthesis. Thank you so much.